Imagine a future where we could rewrite the human genetic code, curing diseases and designing our descendants. That's not science fiction, it's the reality of CRISPR-Cas9. In this brave new world, genetic engineering holds the potential to revolutionize medicine as we know it. Picture a world where inherited diseases are a thing of the past, where each new generation is healthier, stronger. This is the promise of CRISPR-Cas9, a gene editing tool of unprecedented power and precision. But what exactly is CRISPR-Cas9 and how does it work? CRISPR-Cas9 sounds like something out of a sci-fi movie, doesn't it? But it's actually simpler than you think. Imagine life as a grand recipe book called DNA. Each page holds a unique recipe, a gene, that instructs the body on how to create a particular feature like the color of your eyes or your height. DNA is a long, complex molecule found in every cell of our bodies, and it carries these instructions for all the traits that make us who we are. Now, what if you could change a recipe? Perhaps add a pinch of blue to those brown eyes or a few extra inches to your height. That's where CRISPR-Cas9 comes into play. It's essentially a pair of molecular scissors, a tool that allows us to edit the recipes in our DNA. CRISPR, which stands for Clustered Regularly Interspaced Short Palindromic Repeats, and Cas9, a protein, work together to cut DNA at precise locations. Think of CRISPR as the navigator and Cas9 as the cutter. CRISPR guides Cas9 to the exact spot in the DNA that needs editing. Once there, Cas9 makes the cut, and the cell's repair machinery kicks in to fix the break. But here's the clever part. Scientists can provide a new piece of DNA, a revised recipe if you will, that the cell uses as a template when repairing the break. This way, the DNA ends up with a new sequence, a new set of instructions. It's like having a word processor for our genes. With the find and replace function, we can locate a specific piece of genetic code and swap it out for a new one. We can fix typos, errors that cause diseases, or even add new features. But it's not as simple as it sounds. The process is complex and requires a deep understanding of genetics and molecular biology. While we've made great strides, we still have a lot to learn about the potential and limitations of this revolutionary tool. In essence, CRISPR-Cas9 gives us the power to edit life's instruction manual. Now that we know what CRISPR-Cas9 is, let's explore its potential. CRISPR-Cas9, this seemingly inconspicuous molecular machinery, holds immense power, a power that can revolutionize the world as we know it. It's like having the world's most precise and skilled surgeon at a microscopic level. And what's more, this surgeon doesn't tire or falter. One of the most captivating applications of CRISPR-Cas9 is its potential to cure genetic diseases. Imagine a future where diseases like cystic fibrosis or muscular dystrophy are just names in textbooks, no longer a life sentence for millions. By precisely editing the faulty genes, CRISPR-Cas9 could potentially rectify the genetic errors that cause these devastating diseases. But the power of CRISPR-Cas9 isn't limited to human health. It stretches its arms into agriculture too. By tweaking the genetic code of crops, we could improve their yield, enhance their nutritional content, and make them more resistant to pests and diseases. Imagine a world where food scarcity is a thing of the past, where every crop is a supercrop, feeding more mouths with each harvest. And speaking of pests, CRISPR-Cas9 could also help us eliminate them. By manipulating their genes, we could drive certain damaging species to extinction, protecting our crops and our homes. But all these possibilities aren't just dreams, they're becoming reality. Every day, researchers worldwide are making breakthroughs. They're pushing the boundaries of what we thought possible, harnessing the power of CRISPR-Cas9 to create a better world. Just recently, scientists used CRISPR-Cas9 to cure a genetic disease in a living adult mammal for the first time. This breakthrough brings us one step closer to a future where genetic diseases are a thing of the past. With such potential, it's clear why CRISPR-Cas9 is considered a game changer in modern medicine. It's more than just a tool, it's a beacon of hope, lighting the path towards a future where we no longer have to be victims of our own genes. And that, my friends, is the power of CRISPR-Cas9. But with great power comes great responsibility, and CRISPR-Cas9 is not without its ethical concerns. Imagine a world where we could pick and choose our children's traits like hair color, height, or intelligence. Sounds like science fiction, right? But with CRISPR-Cas9, that world could become a reality. This is where we start to grapple with the concept of designer babies. While the idea of eliminating genetic diseases is appealing, the thought of genetically enhanced humans is more contentious. We're talking about the potential to alter the course of human evolution, and that's a hefty responsibility. Then there's the potential for misuse. With any powerful technology, there's always the risk it could fall into the wrong hands. Picture an entity using CRISPR for malicious intents. The results could be catastrophic. And what about inadvertent consequences? Even with the best intentions, we could accidentally introduce new health issues or disrupt delicate ecological balances. But here's the good news: we're not in this blind. We have the chance to establish regulations and safety measures before these scenarios become realities. Oversight is crucial. We need international cooperation and clear guidelines to ensure CRISPR is used responsibly and ethically. And this isn't just about scientists and lawmakers. Everyone has a stake in this. Public dialogue and education are vital to making informed decisions about how we use and control this technology. We must also consider the social implications. What happens when only the wealthy can afford genetic enhancements? It could exacerbate existing inequalities and create a new form of discrimination. These are complex issues that require thoughtful consideration and robust public debate. The potential of CRISPR-Cas9 is immense, but it's a power that must be wielded with caution. We're standing on the brink of a genetic revolution. How we proceed will shape not just our own futures, but those of generations to come. So, where do we go from here? What does the future hold for CRISPR-Cas9? The road ahead for this groundbreaking technology is both thrilling and fraught with challenges. It's like standing on the precipice of a new era, holding a key that could unlock unimaginable potential. But with great power comes great responsibility, doesn't it? CRISPR-Cas9 is not just a tool in a scientist's laboratory, but a beacon of hope for the future of medicine. Ongoing research is pushing the boundaries of what's possible, with potential breakthroughs on the horizon that could transform our approach to treating diseases. Imagine a world where genetic disorders become a thing of the past. A world where we could potentially tweak the genetic code to eliminate hereditary diseases like Huntington's, cystic fibrosis, or even certain types of cancer. The prospects are extraordinary, but it's essential to temper our excitement with caution. The future of CRISPR-Cas9 also lies in the hands of the public, not just the scientists. It's crucial that we foster understanding and discussion around this technology. This isn't just about the science, it's about the ethical, social, and legal implications that come with it. As we venture into this brave new world of genetic editing, we must ensure that the power of CRISPR-Cas9 is used responsibly. We need to establish robust ethical guidelines to prevent misuse. We must ensure that our excitement for the potential of this technology does not outpace our understanding of its implications. And while we grapple with these questions, the research continues. Scientists are already exploring ways to make CRISPR-Cas9 even more precise, reducing the risk of off-target effects. They're also investigating methods to deliver the CRISPR system into the body more efficiently, opening up new possibilities for treating diseases. The future of CRISPR-Cas9 is also intertwined with the future of personalized medicine. As we better understand our genetic makeup, we can tailor treatments to the individual, potentially revolutionizing how we approach medicine and healthcare. The future of medicine is here, and it's spelled CRISPR. As we stand on the brink of this new era, we must remember that the power of this technology is not just in its potential to cure diseases, but also in the ethical decisions we make about its use. The question now is, how will we use this power?